Everybody. I'm Danny. I'm Danny Lasky. I'm a third year neuroscience PhD candidate at UC Davis. Um, yeah, I've been working in the sleep and epilepsy. It's a pretty niche field, the overlap between the two, but I've been working in that even since I was in a previous lab at Wisconsin-Madison. So it's been about like 5 years now. And yeah, I'll share some epilepsy basics and then a little bit about grad school and my own like thesis work and what that kind of looks like.
Nice. And for me, I am Danny's mentee. I've been working in the Fuller Lab along with the Peterson Lab for quite some time, and I'm currently an undergraduate majoring in neurobiology, physiology, and behavior. But together, me and Danny, we work on slow wave sleep along with epileptic mice. So I'm super excited to present today's workshop to everyone. But yeah, let me get my screen up. Please let me know if you're able to see the screen.
We can see your screen.
All right, sounds good. Okay, so welcome everyone to the lecture portion. But does anyone know what this album is? It's from the Passion Pit. But one of my favorite songs from here, it's called Sleepyhead. And if any of you out there know, it's a really, really great song, so make sure to check that out. But getting started with today's presentation. Again, short intro. I worked with the Department of Neurological Surgery and Neurology, and with my two amazing labs, the Fuller Lab and the Peterson Lab. So, a little bit about me and my current research. For me, I started my research into neuroscience fairly early into high school. I always had a passion of neuroscience based on my family's background with our history of like neurological disorders, and also working as a medical assistant, being surrounded with a lot of people with chronic neurological diseases. It definitely brought me a lot of exposure to the different types of symptoms and the types of people that have to deal with these neurological diseases. That kind of exposure pushed me into the direction of research where I got to be more hands-on and get to be more involved with studying about the systemic issues and what causes these neurological diseases to occur. Specifically, my interest lies within Alzheimer's disease, and I've recently gained a huge passion for epilepsy research. And so this is one of the main projects right now, or the main focus of the Fuller Lab along with the Peterson Lab. We utilize slow wave sleep enhancement for neurological diseases such as Alzheimer's, Parkinson's, and epilepsy. So what exactly is slow wave sleep enhancement? First, we have to understand what slow wave sleep is. It's categorized as the deepest non-REM stage, which is the third one. It's characterized by high amplitudes of zero point 0.5 hertz delta oscillations. And I know that that's a little bit confusing, but basically to dumb it down a little bit, it just means that the signals sent out by the specific neurons or the region is around zero point 0.5 to 4 hertz. And that helps induce slow wave sleep. And so, why do we study this? There's a lot of correlation with slow wave sleep, along with the reduction of symptoms such as seizures in epileptic mice, along with reducing the amount of protein misfolds inside of the brain for diseases such as Alzheimer's or Parkinson's. And how we do this is we study slow wave sleep through chemogenetic designer drugs by activating the GABAergic neurons in the parafacial zone. So, a little bit about the parafacial zone. It has these neurons that sends out inhibitory signals towards the wake promoting neurons. These wake promoting neurons project all the way up to the cortex and has a downstream effect, which helps the person stay awake throughout the day. But because of the parafacial zone, it sends these inhibitory signals towards the wakefulness neurons, which then kind of stops the signals being sent, which then induce slow wave sleep, basically helping the person fall asleep faster. So if I'm going too fast or if there's any confusing terminology, feel free to like send a question in the chat or stop me at any time. But that's like the gist of it. So these neurons, they inhibit another set of neurons that project wakefulness, and then that causes slow wave sleep. And along with slow wave sleep enhancement, it also accelerates this thing called glymphatic clearance. Glymphatic clearance basically means when you fall asleep at night, this mechanism helps clear out all the dirty, messy bits inside of your brain. So without proper sleep, this process cannot fully activate. And people, they start becoming moody or protein misfold starts happening inside of the brain, or just a bunch of accumulation of very, very bad bits starts happening. And this is usually one of the early causes for many of these neurological disorders. And so, as you can kind of see with our research, sleep is extremely important for keeping your brain hygiene extremely clean and also helping you function throughout the day. And that's what the focus of the Fuller Lab. Um, this is the Fuller Lab's focus, where we investigate utilizing these slow wave sleep enhancements. But yeah, as you can see here, this picture to the right, we have a viral injection through one of the burr holes in the brain. This right here is a skull, and you can kind of see the hole. The holes we drill, they're coordinated using bregma and lambda, which are little origin points on the brain. And through these burr holes, we inject a virus into that specific hole so that we can create an artificial receptor for our designer drugs to help activate to induce slow wave sleep. And then this picture down here, um, this is showing the effect of the slow wave sleep enhancement. So up here are vehicle designer jellies. They're basically consisting of saline. And so this is what normal sleep in the mice looks like. The red parts are the wakefulness, while the green are the non-REM sleep. And you can kind of see that it's like spread out, looks fairly normal. But then after the slow wave sleep enhancement with the CNO jellies, we see that the sleep becomes more consolidated, where the non-REM sleep becomes much more packed onto this side, while the wakefulness duration is much longer on the right side. So to this, it looks more organized and synchronous. Then moving on, here's another project that I worked on, and this was mainly with help of Dr. Christelle Anaclay. She allowed me to work with these NOxHET3 mice, which basically carry a heterozygous mutation of, the NADPH oxidase 3. Basically, in what this means is that these mice carry a gene there, which impairs the formation of the otolith, and this gives them vestibular dysfunctions. This, vestibular dysfunction kind of messes up their movement and balance, and we were trying to investigate if this dysfunction affects the quality of sleep. And so with this project, I was able to work with these mice and learn how to sleep score. And so that process was extremely, extremely fun because I was able to analyze the different states between wakefulness, non-REM, and REM sleep, and categorizing it with our quantifier program. Next is going to be a more recent project where I worked with Danny. We are conducting a sucrose preference test, which measures for anhedonia in epileptic mice. Anhedonia is basically the loss of pleasure. It's one of the core features of depression and is very common in epilepsy patient. With the sucrose preference test, the mice has to choose between two bottles, which is one consisting of water and the other consisting of around 0.1% sucrose. The main importance of this test is to see if the mice has a preference towards sweet things or not. If the mice is just drinking from both bottles without, you know, seemingly having a preference for something, then it kind of shows that they have anhedonia, which is the loss of pleasure. This is extremely important because this type of research can also be applied towards clinical settings too, where many patients they also um, have similar patterns with their behaviors. And this also has many connections with sleep quality as well. With anhedonia, we plan to connect that with sleep quality, seizure control, and also um, we plan on utilizing this research to correlate it with what we see normally in clinical settings as well. Now, moving on to laboratory procedure. Aside from all the projects that I've worked on, these are the common laboratory procedures expected in the Fuller lab along with the Peterson lab. So I started off doing a lot of PCR, which is polymerase chain reaction. It basically takes specific DNA sequences to confirm genotypes. So we take a little tissue from the mice by either tail snips or little ear punches, and then we put it in an extraction solution. And afterwards, we have our mixture of tissue prep along with our neutralization solution. And we put all that into our tissue to then extract specific strands of DNA. After we extract that specific strand, we can use different combinations of primers so that we can bind with these DNAs. So once we incubate it and we run it through an electrogeophoresis, we're then able to see the bands split apart and travel down the electrode to confirm whether this genotype matches with this specific mouse. This technique is extremely important because many researchers they need to be able to confirm specific genotypes and its correlation to neurological diseases. So, for example, going back to the NOxHET3 mice, um, before we can even start the experiment, we would have to confirm if the mice has the NOxHET gene or not. So PCR, it's extremely important. It's it takes a lot of precision and takes a lot of careful, detailed procedures because usually if you like tap your micropipette on something or you do a step wrong, that can put you all the way back to the first step. So doing the thing takes 30 minutes and then incubating it takes another 2 hours. Then running it through a gel takes another hour. And so if you mess one of that thing up throughout the process, you would have to restart back all the way at the beginning. So it takes a lot of care, but it's a great first initial skill to learn because it helps you practice being careful with reading the protocol, along with helping the PIs and other postdocs kind of assess like your aptitude to learn and also continue to try even if you fail or mess up. But I mainly learned this skill through this program called Big RT at UC Davis. They teach a lot of great introductory research skills, that being PCR, electrogeophoresis, and tissue extraction. So I would recommend that all of you kind of check that out. I will also be sending resources later on into the email so that if you are interested in taking the Big RT program, you have access to that. And they will certify you for lab safety as well. So yeah, that's just another great opportunity. And then going into electrophoresis. This basically is just taking the PCR that we made, putting it into an agarose gel, and running it through, well, first putting a buffer and running electrical currents to separate the DNA bands. And that's great because we're able to take that gel and image it using UV light so we can see the specific bands. Another another common skill that we would have in the lab. Lastly, next is going to be tissue extraction. This is just taking specific regions of the brain, slicing it into small microns, and many researchers, they use this to see if they injected into the specific region correctly, or just seeing like the neuronal population, depending on what procedure that they're performing on. So what I mean by that is immunohistochemistry. Some researchers, they visualize proteins in the brain sections using fluorescent proteins. This are fluorescent antibodies. They do this by having specific antibodies from things such as like mice, chicken, cow, rats, and that would bind to these specific proteins and kind of combine with it and express fluorescence so that researchers are able to scan it through a machine so that they can see the specific proteins in the brain. And all of these skills, they take some time to build up towards, but commonly when you're just joining a lab, you would be starting off with PCR and gel electrophoresis. But the other ones, they're mainly used for neuroscience labs. And then next are the surgical procedures. So these, I would not say normally undergraduates would do, but I had the amazing opportunity to learn from Danny, along with my other PIs, on how to conduct some of these procedures. The first one that I learned was the open heart perfusion. This, we basically slice up the mice, or after we put it down and anesthetize it with either ketamine or euthanol. And once the mice is under, we can check for a toe pinch, which then sees if the mice is able to respond to pain. Once the mice is completely unresponsive to the pain, we would then dissect to horizontally towards around the thoracic. And we do that by clamping on the sternum, cutting a little bit, pulling it up while the heart is exposed, and then injecting a needle into the bottom left ventricle all the way to the top left atrium. And after we do that, we flush it with saline so that all of the heart and the rest of the body pumps saline throughout so that once we dissect the right atrium, all the blood, pushes outwards. So perfusion is basically just pushing saline throughout the body to drain all the blood out so that once we start fixing the brain, it's a lot easier because there won't be a lot of blood getting in the way. And after we have a successful perfusion, we would then switch it to formalin, which then fixes the tissue where we can decapitate the mice and extract the brain. And once we do that, we have the brain, we can move on to our next steps of using the tissue extractions to then either test for protein fluorescence or just slicing it to check if the injection site is accurate. And next is going to be the viral injections. So going back to the picture that I showed before, right here, this would be what the viral injection looks like. It's a glass pipette with the virus itself being injected into the brain. Danny would go more in detail about the specific procedure, but it was a really, really cool thing to be able to see. And next, we're going to be talking about the head plate implantation. So these are just surgical attachments of EEGs, EMGs. Usually we have frontal, parietal, and cerebellum EEGs along with the hippocampal. Danny, he has his 8 channels, while in the Fuller lab, we tend to use 6 channels. So it really depends on which specific region of the brain that you're trying to measure, but each of these specific electrodes are used to measure a specific output. So, for example, the EEG tends to be used to record non-REM sleep along with REM sleep. And then actually the EEG and the EMG are both used to record for REM sleep, wakefulness, and non-REM sleep. Later into the slide, I will show you how you're able to categorize them utilizing the electrical signals sent to the EEG and the EMG. And this right here is usually what the 6 channel looks like. Down here is a picture of the 8 channel, the one that me and Danny uses. But overall, it's a really cool process. You have to like build it under a microscope and you have to be very precise with it. And you have to make sure that each screw is put in correctly, or else, the signal output is not going to be as great. And then all these procedures, they're IUCOC approved. And you might be asking, what is IUCOC? But they're basically this government body which kind of looks into labs and their works with mice and or just animals in general, and they approve whether the protocol is safe, well, not safe for the animal, but ethical for these animals. So yeah, every single protocol here, we would have to submit it through a protocol towards the IUCOC, and then once they approve that, we can continue with our research. Then I move on to the next slide. So, what exactly is neuroscience? So, neuroscience is the scientific study of the nervous system. This includes structure, function, and the diseases. How do you even get into neuroscience? Usually, it starts with research labs, volunteer positions, and sleep study clinics if you're interested in sleep itself. For me, I got through it by talking a lot with the undergraduate research center, taking a bunch of seminar classes to understand how to network, read publications, along with how to write a code email. The undergraduate research center is a very great place to go to because usually they would help you with your code emails along with your resume and also the career center as well. They also do the same thing, but the undergraduate research center has a portal where they're able to connect you with different labs and also give you references to what specific sites are actually open and looking to hire. And after I gained those skills of developing myself professionally, I then moved on towards Handshake. On Handshake, it's a large job listing site for UC Davis students, and you're able to see the postings of many different labs that are looking for undergraduate researchers. The positions are either paid or unpaid, but overall, my main goal was just to get into a lab and get exposed to research. And so um, after looking for a specific research that I was interested in, which was Alzheimer's disease and other neurodegenerative diseases, focusing on sleep or early prevention, I came across the Fuller Lab, which then I applied to. And after applying on Handshake, I decided to send an additional email towards the lab manager, expressing that I've read their publication, I'm extremely interested in their research. I want to contribute this amount of hours along with this is my topic of interest, and just sincerely telling them like how interested I am in the work that they do. And after I'd done that, I got booked for an interview, and from there it was just getting like really far into research just by reaching out, just by taking that initiative. But yeah, I encourage every one of you to also do the same, where you just take as much initiative as you want. Don't be afraid to embarrass yourself because, most labs, they don't really expect you to know what you're doing before you actually get into the lab. So they're willing to train you and help you. All you have to do is just ask them first. But back to the presentation. Many key skills for neuroscience is just curiosity, basic biology, chemistry, data analysis, just like, for example, R Studios, Google Sheets, or other programs that you kind of use to quantify data or compute things very quickly. That skill is very useful if you're working with like really large data sets. And then also wet lab techniques such as like PCR or you know, other skills such as micropipetting, just anything that can be extremely useful into getting into neuroscience. And so the my kind of rough timeline was just getting a topic of interest. What are you interested in neuroscience? Then finding a professor through the department. You can search this up by just going onto the UC Davis website, looking into the departments, and reading through the professor and their researches. After you read through their publication, just send a cold email. And yeah, that was just my process. And so let's talk about the undervaluation of sleep. Why does it matter? And commonly today, why do we undervalue it so much? So sleep is often sacrificed in modern culture, and it's treated as a luxury rather than a biological necessity. Public health, we rarely emphasize sleep as a preventative health measure, mainly not because of anything of stigma or, of that matter. It's more of just the lack of understanding behind sleep and its importance. So, early intervention in sleep habits may delay or prevent neurodegenerative disease onset. This, the type of research that we do in the Fuller Lab and the Peterson Lab have shown that the amount of sleep that you get, along with the quality of sleep that you get, do in fact help with these symptoms. And so, it's extremely important to be able to understand how sleep works, along with how it affects your body. And then the lack of sleep research in higher academia. Um, the thing about the neuroscience field and sleep itself is that it's kind of niche. Not a lot of people investigate it as much as you would think. And this has many opportunities for people to discover new things, but it also has a con of being very, very niche to understand. Not a lot of people understand about it. And so being able to conduct this type of research has been extremely meaningful for me because I'm able to show how much sleep can help the body along with how much it can help you in the future. And so moving on, sleep and neurodegenerative diseases. What is the connection? Chronic poor sleep is linked to increased risk of Alzheimer's disease, Parkinson's, and epilepsy. Sleep disruption accelerates the accumulation of misfold proteins. So there are these proteins in the brain called beta amyloid proteins and tau proteins. And what happens is without proper sleep, the lymphatic system, which is the cleanup process, it doesn't work as well. And so these proteins, they start accumulating and binding up together and plaquing up, and all of these start floating around the cerebral membrane. And this causes a lot of issues, such as internal skeletal deterioration, along with just starting the onset of like early dementia or making it harder for your neurons to fire and send signals between each other. Having good quality sleep helps with this and helps with the clearing up process. And so let's talk about the biology of sleep. So, what is sleep exactly? Sleep is reversible. It's a periodic state of reduced consciousness and motor activity for survival. There's two main stages of sleep. This consists of REM, which is rapid eye movement, and non-REM, which cycles every single around 30 minutes. Non-REM includes slow wave sleep, which is the deep sleep. So there's N1, N2, and N3. And slow wave sleep is categorized as the N3, which is the last one. And it's critical for memory consolidation and lymphatic clearance, which we've discussed before. REM sleep is associated with dreaming, emotional regulation, and synaptic pruning. And healthy adults usually require 7 to 9 hours of sleep, and sleep depth accumulates overnight if you're a person that tends to sleep much farther into the day and in the daytime you decide to take multiple naps to try to compensate for that, thinking that you got your 7 hours of sleep a night and you're good. That doesn't actually work because sleep depth accumulates even with that. And it also pushes your circadian rhythm much farther into the day, into the next day, which makes it even harder for you to fall asleep. So, getting that full one cycle of 7 hours of sleep at night is extremely important. And knowing your chronology of when your body needs that rest is extremely, extremely important. So don't try to do any brain hacks where you're trying to compensate for your sleep by taking a bunch of naps or just like, thinking that you can sleep for 4 hours and then work for 4 hours and then sleeping another 4 hours. Those things usually do not work. So just make sure that you know what your body needs. Then moving on. So here's the categorizations of the three sleep stages. We have wakefulness, non-REM, and REM sleep. So we see up here, wakefulness has a bunch of activity down here. This is where our EMG is. The EMG shows the muscle movement, and the EMG is usually located under the neck muscles, which has very, very, I guess, crazy looking signals. That's the easiest way I can describe it. And the EEG, it's very low amplitude, but still, it's a bit messy. But it's in contrast, for an EEG of the non-REM sleep, it's categorized by very high amplitudes. And you can see that, the waves or the signals are much more apparent, like they're much more spiky. Well, then the REM sleep, it's supposed to be more harmonious, where, well, it doesn't really look too harmonious here, but usually the EEG shows a very harmonious movement, while the EMG for it has very, very low amplitudes. So you're like barely seeing anything on the EMG because when you're sleeping in REM sleep, it's categorized by muscle tonia, right? Which means that there's no muscle movement whatsoever. So that makes sense with the EMG. If you see muscle movements while you're having REM sleep, what do you guys think could be the cause of that? Any guesses? Any guesses? Feel free to send Any guess in the chats? Give you a minute.
Wow, do you mind repeating the question? This is Emily over here. Sorry.
Oh, yeah, of course. So the question is just if you're able to see activity in the EMG, then, but this is also in the REM state, what could be the reason for that?
Is it because when we're in REM, that's also kind of similar of wakefulness and when we're dreaming and we're moving around?
Yeah, yeah, that's correct. So patients with like, uh, I guess people who like sleepwalk or usually like in dogs, like while they're dreaming in the REM state, they tend to twitch a little bit, and that can be picked up in the EMG. So when you sleepwalk or you have like those little muscle twitches, they're usually picked up by the EMG signals. So yeah, great guess. Moving on. So now let's talk about the chiasmatic nucleus. This is basically a circadian clock, or a clock in your brain that regulates your circadian rhythm by a 24-hour biological cycle. And then this also, once it takes in the light signals outside, it then helps your body send melatonin, and this helps you fall asleep. Then adenosine, it's basically a hormone, basically acting as sleep pressure. As the day progresses, more adenosine accumulates, and it's cleared once you fall asleep. So this would help you fall asleep as well. And this is kind of a diagram of what it looks like. But light goes in, gets interpreted by the suprachiasmatic nucleus, and then this regulates your circadian rhythm and sends hormones throughout the body.